Well, students, that was a short commercial break, and we shall continue with the spotters. Describe. Whenever you see a slide, don't panic. Always look for a lining epithelium. In this case, I am finding some columnar cells and goblet cells. Here it looks more like squamous. We used to call it transitional epithelium, but then a few people can write columnar epithelium and goblet cells, good. This is the subepithelial stroma. It is looking edematous. Look, the cells are all getting separated, therefore I call edema. But more important is there are some well circumscribed or circular lesions that I can make out. And same old friend of ours, these dark cells are all the inflammatory cells, the lymphocytes. So it is a fragmented lesion because they will be removing it in fragments, lined by ciliated columnar cells and goblet cells. The underlying stroma is edematous, and I'm finding these are all called sporangium. They may be full, they may be empty. Here I'm able to see a few spores. This is almost empty. And this is another one. So it is a sporangium that is completely filled with these endospores. The stroma here is again edematous with a lot of these inflammatory cells. There is an epithelium that is covering the tissue. So it is a case of rhinosporidiosis. This again looks almost similar. This is the rhinosporidiosis that we had described recently. But then look at this. There is again an epithelium. It can be a pseudostratified columnar epithelium, but then the subepithelial tissue is edematous. Look at this lot of empty spaces that we are seeing. That is because of edema. There are a lot of blood vessels also. Again, our friend, look at these numerous dots. They are all inflammatory cells. It can be lymphocytes or eosinophils. In this case, it is lymphocytes. Yet another case of the same. I am finding that there is a ciliated columnar epithelium and the goblet cells. Pseudostratified, it can be lymphocytes or eosinophils. Mucus glands sometimes can be present. And dear students, please be careful. There won't be any ulcer or anything. Why I am saying this is there is another slide of ulcer with granulation. We will be mistaking it for this. So it is an inflammatory polyp. The previous polyp was rhinosporidiosis. Look at this particular slide. I would like you people to see again our friend. There is a stratified squamous epithelium. Very few lesions are like that. So you should be able to number. In this case, there are no rete ridges. It is flattened out. And there is somewhat an acellular zone compared with this that I can see. No dermal adnexae are made out in this. Another slide that we saw was the keloid. Again, that was having epidermis, no adnexae, but in this case, it is full of cells. Again, look at this over here. There is the melanin pigment, the stratified epithelium, there is the keratin, and below that, lot of cells with these holes or vacuoles. That is there, and there is an acellular zone. Acellular zone below the epidermis called the Grant zone. Sheets of foamy histiocytes, no granuloma. And a diagrammatic representation of the same, the epidermis, dermis is packed with these foamy macrophages that are arranged in sheets. Don't draw a granuloma. So this is the acellular zone. It is a case of lepromatous leprosy. Look at this particular case on the contrary. There is an epidermis. This is the rete peg or the rete ridge. So again, rete ridges are seen in this. In this case, multiple circumscribed lesions are seen. Sometimes they are joining, but I am able to see a giant cell also. At a higher magnification, this may be focused. I am able to see a Langerhans giant cell no necrosis. So it is again the same picture. What do I see? There is a skin, non-caseating granuloma, epithelioid cells, lymphocytes, and Langerhans cells. They are mostly lower down periadnexal. The epidermis is unremarkable. The diagnosis is tuberculoid leprosy. Describe. So this is a favorite one of mine. I am finding a structure that is quite irregular. I used to describe it as a holy leaf pattern. There is a thickened area over here, which I call a chitinous layer. And then there will be a wall, and this is the host tissue that I'm finding. So this is again the same. This is the head of the scolex that is developing. So I'm finding particularly the lower part, it will be having this leaf-like configuration or a holy leaf pattern. Look at this beautiful picture of it. A holy leaf-like pattern is there, and this is the gross picture of a cyst that has been removed. There is a space over here. So there is an endocyst. This is the endocyst. This is over here. Ectocyst is the surrounding tissue or the wall of it. Pericyst is the host tissue. And there is a chitinous appearance. There is no epithelium over here. Holy leaf-like lumen. Scoliosis, that is the hooklets, may or may not be seen. No epithelial lining. And the diagnosis is cysticercosis. Another favorite one, very easy, multiple sheets of cells, more or less uniform, empty looking cytoplasm, hardly any material is seen. And externally it is covered by a capsule. This will be focused in the exam. So look at this one. This red line is the capsule. And these are all large vacuolated cells. And in some I can find peripherally pushed nucleus. So these are all the features of it. No pleomorphism, no hemorrhage. The diagnosis is lipoma. This is again a very easy slide for you. You find that they are in bundles. And compared to the previous one, I find that the cells are all long. All the eosinophilic material, pink material is the cytoplasm, and the blue material is the nucleus. So bundles of spindle-shaped cells that can be arranged in a whorled pattern. Nucleus at a higher magnification, it will be having a cigar-shaped appearance. It is a leiomyoma. Another slide also may be kept. Compare this with this. So this is also a leiomyoma, but I am finding some acellular pink material in this. So what can it be? So this is one. Tumor cells in bundles, spindle-shaped cells, and whorled pattern. Nucleus will be blunt and cigar-shaped. Okay, no pleomorphism, and homogeneous eosinophilic acellular areas are there. So compare both. This is the previous one and this is the present one. Plot of this pink acellular material, and the diagnosis is leiomyoma with hyaline change. So this is again a tumor. There are large cells and these are intercellular bridges. Look at this over here. So this is a nucleus. Some are pale, some are dark, and in some region they tend to be forming a more eosinophilic material. It looks like keratin. So this is a better one. I am finding tumor cells in nests or in sheets, and there is a concentric arrangement. Multiple nuclei are there. So this is an epithelial pearl. So this is what you people see. Dark areas are epithelial pearls. Pleomorphism can be there. I would like you people to kindly describe this as I have given. The diagnosis is squamous cell carcinoma. So towards end, a treat for all of you. I have opened a pack. I am finding that these have got no relation to each other. Obviously, these are assorted biscuits. They can be sweet, salt, or cream, or what wafers are here. So these are called assorted biscuits. That is exactly what I am finding in this slide. There are spindle-shaped cells, there are some glands, and some are actually a neural tissue. In this one, there are glands. These are again adipocytes that I can make out. This is in between a stroma. So numerous, absolutely no relation to each other. That is exactly what I am seeing over. This tumor has got representations of the ectoderm, endoderm, and mesoderm. So I'm finding a stratified squamous epithelium. This is cartilage, connective tissue, glands are there, etc. So it is a teratoma. See this again, cartilage. You find this in a blue background. I'm finding vacuolated cells. These are some kind of sebaceous glands. This is a fat tissue that you already see. Remember the lipome and connective tissue, the pink bundles of cells. So all these are seen. So you can describe all this. The diagnosis will be a case of teratoma. This is another one. I find that there is a lesion that is projecting above the surface. This is a normal mucosa. I am able to see empty or the goblet cells. These are some kind of columnar cells. This is normal, but this fellow is projecting above the surface. So a lesion that projects above the surface is called a polyp. There can be a stalk and there can be a lesion as such. So what am I seeing in this? Lesion is projecting above the surface, well punched out glands, as though with a punching machine we have punched out. And the lamina propria is the surrounding supporting tissue. The glands are lined by columnar epithelium. It is well differentiated, so it is a tubular adenoma or a polyp. This again, this is important for us. You would have seen tuberculous lymph node. I am finding this bluish material not very clear. And this is a capsule, pericapsular fat. But below the capsule, there are multiple irregular glands that I can make. So it is a metastasis. And look at this again. Here I can make out the lymphoid follicles quite easily. There is a capsule, and in the subcapsular spaces, there are irregular tumor glands which are. So this can be an adenocarcinoma which is metastasizing, or you can have whorls of keratin and polygonal cells which can be a squamous cell metastasis. Anything can be kept. So lymph node with capsule, lymphoid follicles, subcapsular deposits, solid or glandular. So you should not write both. You must write what the case is. It is a lymph node metastasis. That is what you people are seeing on these pictures. The moment you see the glands and then the lymphoid follicles, the diagnosis is quite easy. Lymph node metastasis. Question that.